In order to obtain the foreign gene product, the transformed host cells are usually grown in a specialized container known as the bioreactor. This bioreactor it's where the all optimum growth condition is provided along with the nutrient to the particular targeted cells, which will be growing there and producing all the targeted proteins or whatever chemical agents that is required. Now, a small scale unit is the one that is shown in the picture here now, and in that you can see that the central unit is actually the place where it is all grown. The liquid is red in color here, the nutrient broth in which the organism is growing, and all supporting instruments are there working along with the particular apparatus so that the apparatus can run properly. And it is always that the thing is regulated and controlled. This unit is only 10 liters maximum, and the one that you are seeing now is actually the one that is around 100 liters of capacity. Now everything is computed and everything is programmed such a way that it will run all by itself. Now usually the container that inside which this is all done, they are kept sterile and clean before the entire process has been initiated, and then the broth is added along with that culture media which will contain the targeted host organism. Now, once they both things are there inside, the targeted organism will be allowed to grow there and properly utilize all the nutrient components and produce the targeted protein. So when all these targeted proteins are produced, it will be later on taken out from the entire setup through that effluent pipe. Like there's a tap system is there through which it will be all it will be all sorted out, it will be all taken out, but it is all allowed to grow right there. Now while growing, it is definitely going to produce a lot of heat, and this heat is going to be regulated by certain sort of cooling jacket that is there around it. And this cooling jacket, or sometimes it's a hot jacket also, is a thermal jacket which will regulate the temperature some way somehow, as necessary. Well, one thing should be mentioned here, that entire setup is completely automated. It is controlled by certain instrument setups and everything, and they contain several kinds of probe by which it can be all monitored. Even the temperature that we're talking about here, the temperature is regulated or it is monitored by a probe which is shown here, and the probe checks the temperature, and then necessary treatment is done. If it requires heat, then heat is provided. If it needs to be cooled down, then it will be cooled down as necessary. Now, most of the time, the heat produced by the metabolic activity will be enough to heat up the entire system, bringing it to the optimal temperature range. But many of the time, the external influences are required. For that, the thermal jackets are arranged into different ways and patterns. In the first case, we can see here that it is simply the microorganism itself is responsible for the heat and everything. Whereas in the case number two, we can see it is like there is a coil, a jacket or a thermal jacket is actually placed around the entire apparatus, and that thermal jacket can be filled with hot water or can be filled with cold water just to regulate the temperature as necessary. Now it can be containing like some sort of heating coil as shown in like C and D condition, in which this two coils can heat up the mixture and everything. But one thing should be kept in mind, only in a small scale production, means a production which is in the range of maximum 20 liters or 50 liters, that it can be maintained like this way. But anything that is larger, it cannot be done the same way. For that, an external heat exchanger is actually used to cool down the system. Now, when we talk of the rest of the apparatus, then it will have a pH balance setup. So the pH probe, just like the temperature probe, is going to check the pH of the solution, and it is going to dispense as necessary. If any acid or any base is required to be supplied, it will simply automatically dispense that. It is programmed such a way because the entire system runs in a computational system, like it's completely automated. Now, along with it, we will find that there will be a agitator system connected with a motor. Now, this agitator is like a long tube containing terminal sparge, terminal bafflers, where there are this rotating bafflers are used, and these baffles are responsible for agitating the entire mixture. This is necessary to distribute the temperature or distribute the heat throughout the entire system, plus proper access of nutrient with every single microorganism that is growing there or any cells that is growing there. Because If it is not done, then the cell will aggregate at one corner, and only that particular part of the nutrient will be utilized, or the nutrient mixture it will be utilized. Rest of the portion, nothing will happen. So that's why the proper agitation is required, because entire thing should be properly utilized and the production should be gained properly. That's why this baffle, it actually rotate all around the thing all the time, creating an agitation. Along with it, there will be supply of sterile air, because some system they require aerobic condition, and to provide that aerobic condition, air needs to be supplied through the entire thing. Now air, it is usually supplied through this particular section, as you can see, where the air supply is being carried out, and the air will be definitely sterile, because a sterile condition has to be maintained here. Pure contamination free growth of the organism is actually targeted most of the time. So the sterile air supply will keep on providing the sterile air or oxygen to the system, and that will help in proper yield of the entire whatever protein has been targeted. The sensors and all these things, they work all around, all together, and totally this entire bioreactor, it keeps in a sealed contained system, in which it can work properly. It will do the entire growth of the organism. It will help in the growth of the organism, and finally help in production of the whatever protein was targeted, which will be taken out through the effluent pipe, and through that the entire thing will then undergo a downstream processing system. Here you can see in this picture that a continuous exponential growth curve is actually maintained in that particular setup, where the nutrients are continuously fed through the feed pump into that particular bioreactor, whereas the bleed pump is taking away any waste material that is being provided, or the used up or spent nutrients that is being produced there, that has been there. And at the same time, there is this effluent one, which is taking up all the things out, and the filtrate is taking, which contains the desired protein. That is how in the industrial level every single bioreactor works. Now, aeration is a very important part in the entire growth of organisms there, and it is the oxygen which matters for aerobic culture. Now, whenever we talk about oxygen, the dissolved oxygen is going to be very hard for the organism to get it from the solution, because in a solution state, the dissolved oxygen will not be easily available or easily readily usable for the microorganisms. So for that, a sparger system is used, which will supply all the gases through very tiny pores, and in a pressurized manner, where the oxygen molecule will be arranged in small bubbles, and that bubbles will easily dissolve with the entire mixture, making it properly aerated, and that aeration will help in proper growth, or very profound growth of the organism in that particular growth medium. So that's why this sparger tank is used many of the cases where some large scale industrial growth is being done. That time this sparge system is actually used for better oxygen transfer to the entire broth or entire liquid nutrient mixture. Now, when we talk about downstream processing after this, because that is very important and crucial, so it's mainly like after the fermentation, or the bioreactor, whatever is happening there, mostly fermentation. So whatever things are produced, they are centrifuged to remove the cells, or sometimes they are filtered also for the same purpose, and then they undergo some sort of chromatographic technique to precipitate the protein out. So once this protein is being precipitated, they are chemically purified, or maybe even chromatography like ion exchange chromatography or affinity chromatography is used to extract the entire proteins out, specific proteins. Then they are suspended in a solvent and solution, and finally this purified protein sample, or the semi purified protein sample, whatever it is, they are made ready to be sold in the market. They are sold in the chemical market for whatever analytical purpose or whatever it is. Now, depending upon the need, they have different ways of treatment is going on. So it can be like for simple animal feed, if it is going for, then that cells will matter. So the cells will be cultured, by simple centrifugation, it is separated and cells are provided there as a growth medium. Or if it is analytical purpose, then as I said, it is all done in that particular way. It is therapeutic, means in pharmaceutical industry, they undergo even more stringent process of
Sterilized, and then
purification,
finally
and
they are
ultimately the final
processed or
product is
even
being purified to the finest level, and they are homogenized, and sometimes they are added with some adjuvants which will be allowed to properly distribute it through the body system, and then it will be this product will be finally used for pharmaceutical purpose. It can be this antitoxins or vaccines that are there. They are produced by this particular way and by this particular method. Now mostly this bioreactor is used for pharmaceutical industry. It is used for fermentation industry, like alcohol or alcoholic beverages in production and all, chemical production, enzyme production. There are so many diverse places or like where this process or this bio instrument is used.